Hi, I'm Jay Phelan. Let's learn some biology. We're going to talk here about a tool that geneticists use called a test cross. And it's very low tech, and it's not even something that you'd necessarily need to do or have to do ever, but it's still important and useful to understand it because it will reinforce our understanding of Mendel's law of segregation and how the whole process of heredity is working. So we'll start with this question that if an individual is heterozygous, we know that they're going to show the dominant phenotype for that trait, but we don't know what their genotype is. And this was an important thing that first motivated Mendel, was that there wasn't a one-to-one correspondence between your phenotype, what you look like, and your genotype, the hereditary factors that you carry. Sometimes you might be homozygous dominant, you have the dominant trait. Sometimes you're heterozygous, you have the dominant trait. So you don't always know. So for this one, I'm going to use an example that's wrong, that's not true. And I hope that you won't take away from this any false knowledge, but I think it's useful to use as an example. It'll make it easier to learn. Eye color. Now, we now know that eye color is not inherited as one gene with a big B for brown and a little b for blue. It's complicated. It's probably 3 genes, maybe more genes, multiple alleles, but it still mostly behaves as if 1 gene and 2 alleles are coding for it. So we're going to pretend that that's how it is inherited so that we can look at this. In actuality, there's a whole range of dark brown, light brown, grayish blue, and so on. But let's say then that we have someone who just wants to know. So it's a practical issue. So I have brown eyes, and I want to know, am I heterozygous or am I homozygous dominant? Well, we could look at my parents. If either of my parents had blue eyes, then we'd know that their genotype, little b. So they would have had to pass on 1 little b. Then I'd be heterozygous. Unfortunately, both of my parents have brown eyes, so we don't know. So then what else could we do? How could we figure it out? You might say, oh, well, sequence the eye color gene. Well, we don't always have that, and it's expensive and time-consuming. Is there an easier way to figure out a genotype when you just see the dominant trait? And the way that we can look at this is this. So we want to know my genotype for this hypothetical gene of eye color with a big B and a little b. And then we want to know what cross could we do that would allow us to figure out definitively between homozygous dominant and heterozygous. So we know that my genotype has to be big B, or big B, little b. So how can we figure it out? Well, if, in the interest of genetics, I were to go out and find somebody who carried the homozygous recessive genotype, so when I had blue eyes and a little b, and produce a kid with them, what do we know about that cross? Well, we know this. We know that the cross, if I am homozygous dominant, would be big B, my genotype, times little b. And because of Mendel's law of segregation, all of my gametes are going to have a big B. All of her gametes are going to have a little b. Every time they come together, they're going to be heterozygous. What are they going to look like? The outcome is going to be that every single individual would show the dominant trait. In this case, all the kids would have brown eyes, no matter how many we made. On the other hand, remember, we have 2 possibilities for my genotype. I might be big B, little b. If that were the case, then the cross is big B, little b, times this, what we'll call the test cross individual, little b. So she's still going to contribute a little b every time. So every one of the offspring is going to have a little b. But now what am I going to contribute? Half the time a big B, half the time a little b. So 50-50. So if this were the case, can I produce a blue-eyed kid? Yeah, because I could give the little b. In the other case, I couldn't. So I could go out. So let's say we have a kid. I find this test cross individual. We have a kid. And the kid has brown eyes. Do we know what my genotype is? No, we don't, because we already know I have at least 1 big B. So maybe that was the gamete carried one of those, and I pass that on. So how do we figure out what my genotype is? We'd have to have another kid. right? So we have another kid. What if this next kid has brown eyes? Do we know what my genotype is now? No, because half the gametes are going to have the big B. So we have another one. That one could have brown eyes. What if we had 10 kids in a row and they all had brown eyes? Would we know? No. Could have just been luck of the draw that 10 times in a row, even though it's 50-50, we got the big B. We wouldn't know. I'd start to be suspicious. I'd start thinking, I think maybe the only allele that I am passing on is big B. But I wouldn't be certain. On the other hand, what if we produce a blue-eyed kid? Well, then we know my genotype, right? Because that means the kid has little b, which means we already know the test crosser. She's given a little b. I had to give the little b. So in this case, here's a picture of Julia, my test crosser. She has blue eyes, and that's our first son, Jack, who also had blue eyes. So we could stop then. We didn't, though. But in this cross, where you have the heterozygous individual with the test crosser, half the time you're going to show the dominant trait, half the time you're going to show the recessive trait. Now, when you think about humans and kids, that seems ridiculous. Like, you wouldn't want to do that. It would take too long. It's time-consuming, tuition, it's hard. But for something like eye color in fruit flies, you got red eyes, you have white eyes. When you cross fruit flies, you can have your test crosser with the white eyes, homozygous recessive condition. They'll produce 50 or 60 offspring at a time. Now, if the one that shows the dominant trait and we don't know the genotype was homozygous dominant, all 60 of them are going to show the dominant trait. On the other hand, if it's heterozygous, half will show the dominant trait, half will show the recessive. Maybe not exactly, but it's going to be, 30-30 or 40-20 or something like that. But it's very easy. You just look at the flies that come from it, you're like, now we know what the genotype is. So in situations where you're using an organism that you can produce a huge number of offspring, very quickly you can tell what the genotype is of that individual showing the dominant trait that you don't know their genotype. And what's cool about that is that you've now just figured out a tiny, tiny little thing. What is their genotype without having to look at it? So you just have been able to deduce that. And that's why the test cross is super valuable. You can figure out this thing, this invisible thing, what's your genotype, just by looking at something visible, the phenotype. And then you can tell the individual showing the dominant trait, are they homozygous dominant or are they heterozygous?